Back in the day, if you were sick, they would just lead you and toss you out back. But now with artificial intelligence technology, we are able to model human biology and all of biology and maybe make good changes for it. And so there are 3 parts to understanding this puzzle, kind of like a thesis, antithesis, synthesis. The first piece is what's happening and why it's amazing. The second piece is why it sucks. And the third piece is what should actually go down. Okay, so let's start with what AI can do. AI fundamentally, just like it can understand the language of humans and produce documents and help you out on ChatGPT, you can use those same transformer models to understand genetic sequences and what is likely to come after one another. And you can also start to use it to understand proteins and how they're likely to interact. And you can move up the chain of biology to think about how cells might interact. And so we're using this with things like AlphaFold. So AlphaFold's like the simplest, best example where it's like, yeah, AlphaFold 2 was able to look at the structure of the universe and was able to take the number of proteins, 200,000 proteins in the protein data bank. We now know how proteins in general are created. Let's make 200 million of them. And so they're able to look at for any given amino acid sequence, you can think about what that folds into. And then AlphaFold 3 was even crazier because then you could say, wait a second, let's not just think about how one folding occurs, but let's look at how they might start to interact with each other. So these are protein-protein interactions, PPI networks. And you can also use them to understand how the small molecules might interact. And then this one looks at, these simple yellow sugars in yellow. So much of cells doing their work is sugars getting onto them and little small molecules interacting with these bigger structures. And now we can start to model that. And now though, we're moving up to single cell resolution. So we're starting to create these virtual cells. The CZI people have been doing a great job of this. And so this is Tabula Sapiens, which looks at, it's a human cell atlas where you get these 1 million cells and you can start to see how they might operate. That's combined with all these other ones into this bigger platform that has these 100 million unique cells. And in these systems, you can start to modify them and start to try to get them to see their expression pattern. I love thinking of this as a transcriptomic profile where you look at, of the 20,000 genes that humans have, which ones are currently being expressed at any given time. The other folks that have been doing great work here are the Arc Institute people, for example. They have this Tahoe 100 million rather, which is the largest single cell data set. So this one looks at all these different cells. And then when you start to perturb them, when you start to give drugs to them, what happened? And this one is kind of like the Google missense model, where you look at, there's 9 trillion base pairs in here, there's 6 billion from humans, and then 1,000x other ones. What happens if you add or remove these genes or change the base pairs, and you get to see what bad things might occur? Then you got the people at Future House, which are doing the scientific reasoning model for chemistry. And here's this great post by Andrew White from Future House about it. And he's talking about creating a new verifiable function that then these foundational models can act against, where they say, hey, we want to create these molecules on the right. You need to do some stuff on the left side. You need to start to combine things together, things that are purchasable, that will actually work together to make the molecule on the right. And it took them a bunch of time to create this because they need to say, okay, at the beginning, the model was hallucinating and it was thinking of non-purchasable things and it was kind of reward hacking in all these different ways. And as you start to minimize the reward hacks, then what you end up with is something that can repeatedly answer the test question correctly. Just like they can now do all the PhD level questions and GPQA and math and, IMO Gold and all that stuff. You can do similar things, but for any verifiable domain. You just need to define that verifiable domain well. And one of Andrew's learnings is right here, which I super agree with, which is specifying good reward functions may now be the majority of the effort. That's what so much of AI is these days, is saying, hey, we want to get some output in a verifiable domain like math or chemistry or computer science. And so we're going to make a well-defined output. And then we're going to let you, little creature, do reinforcement learning to create that output. So blah, blah, blah. This is to say that in the future, many things like drug discovery and the drug pipeline and understanding how our cells will react to things, understanding different genetic mutations, that those will hopefully be all modeled by these systems where they're able to know how to fold the proteins right. They're able to know the protein-protein interactions. They're able to start to understand how a given cell changes given a certain set of signals from the outside world or a certain set of drugs. And so then we can know a given molecule's transcriptomic profile at any given time. So what are the set of mRNAs and proteins that it is then starting to produce? So you can get the whole proteome, the metabolome, the transcriptome. And once we have those things modeled, then we can start to, in theory, predict how these cells will operate. And if we're able to do that, then we can vastly speed up clinical trials and things like that. So that's the optimistic case. But now I want to go talk about Derek Lowe, who writes this great blog called In the Pipeline, and he's skeptical. He's skeptical, and he might be right. So I'm going to show 3 pieces by Derek Lowe. And Derek is a pharmacologist and a drug designer. And one of the funny things he says is 85% of drugs fail. We have these great ideas, and then they go through clinical trials, and either at the animal level or at the human tox level, they don't work. And he says he's done it for 30 years, and he hasn't had a successful drug yet, even though he's like super, super smart. And so this stuff is just really hard. And so he's kind of skeptical of all these outlandish AI claims. So here's one of his 3 articles that I want to share. The first one is Predicting New Small Molecule Binders. And this is in 2022, August 2022. And he's like, oh, here's this cool competition. We're going to have these 3 different protein targets. Here's all these different chemical species, and you get to explore all the different chemical space here. And maybe you can make these machine learning models, and some of them might have an impact here, and you might be able to find some good new binders or what have you. And what he found out, 2,000 people gave this a try, and essentially what they found out is that it was no better than chance. Even though they had these machine learning models that could explore the space, they had really good data, it was like, no, these machine learning models weren't able to predict some of these bindings better than chance. I think my favorite piece from him is this one called AI in Biology. He's like, I wish I shared Alex Tabrock's optimism here. He says that AIs with access to vast databases of genes, proteins, and networks, and so forth will enable new simulations and learning that has already happened with protein folding. He's like, yeah, that seems interesting, but I'm skeptical. He's skeptical. It's all based in data quality. He's like, yeah, we had this great protein data bank, but there's not other ones that are like it. The biology literature as a whole is not even remotely close to this state. The PDB was amazing. Just like how ChatGPT is a reflection of the internet, 100 trillion tokens on the internet, boom, we can create documents, we can create videos, we can do all that because we have all this amazing data. Our data on biology is not that good. And the only data that we had that was good, this protein data bank, we've kind of used it all up for AlphaFold 2. And so yeah, data quality is an issue, data quantity is an issue, there's not enough data, type of data, protein stuff, there's all these very specific linkages and all these things, and then data completeness. And so I think, as he says, for these reasons, I unfortunately do not expect any big AI-driven breakthroughs in biological understanding anytime soon. What advances we might see will also stick very close to the areas where the above problems are mitigated as much as possible, and that leaves an awful lot still out there. I do agree with this, which is that fundamentally these systems, the AI systems, are a function of their data. Data in, data out, And so we need to find better ways to give these AI systems data if we want them to predict actually novel things. And then his final one from April 2025, so more recently, again, he's like, last night the CEO of DeepMind, Demis Hassabis, appeared on 60 Minutes and said this: proteins are the building blocks of life, everything's firing, 3D proteins are more complex, DeepMind did 200 million in a year, which is true, and now he's doing it with drug development, and he's like, okay, great, it's going to be, usually it takes, 10 years and billions of dollars to design a drug, maybe we can reduce that down to months or weeks. That sounds incredible today, but that's also what people used to think about protein structures, and it would help revolutionize human health. I think one day maybe we can cure all disease with the help of AI. The end of disease? I think that's within reach. Maybe within the next decade or so I don't see why not. And again, you can imagine, what is our boy going to say? What is Derek going to say? Let me be one of the people to say, why not? He's like, yeah, this isn't going to work. And then he just talks about the receipts. He's like, here's like 20 posts I've written that say why this is going to be a really hard thing. And so he has 3 big pieces on this. One of them is what people are calling AI is machine learning. A bunch of the data stuff we've already been doing. 2 is that it doesn't create any new knowledge. It mostly just reproduces. It is a kind of a lossy dictionary for the existing knowledge that we feed into it. And then, it's a compression of that knowledge in a really cool way, but it's just a lossy compression. And then finally, he's like, I strongly believe that all of machine learning is done by looking for patterns in language. And as Wittgenstein said, in any language, there are things that cannot be said. And so for biology, he thinks those things are these important questions. Which of these proteins is more responsible for this particular disease? Which of these proteins would be more likely to lead to a successful drug? And which of these proteins should we avoid working on because such projects will have more potential pitfalls than the others? Boy, these would be great questions to answer. But a machine learning program that knows protein structure cannot answer them, no matter how amazingly well it knows protein structure. And yeah, should we believe that now we can model these systems and so we're going to be able to develop all these amazing new drugs? Or should we believe Derek Lowe, who's like, these systems are not able to produce new knowledge. These systems are too complex. They don't have the right data. Well, I think this part of the answer to this is, at the beginning, what AI is mostly going to do is it's going to do pre-work problems. So it's not going to help that much with drug discovery. But what it will do is something like this, where you can see the growth in physicians and administrators, where from 1970 through 2010, we got all these admin people. This happened in education too. And so with all those admin people, 20% of medical costs are for admin. So it's like, if you can just have less admin, maybe we can save a ton of costs there. This is the idea of things like Alpheus, which is this great platform that does, it's this ambient conversationalist that is hanging out with you. So you don't need to chart as much, and you can just kind of more than describe ambient AI as a true partner in clinical facilitation. That's legit. And so these AIs are going to try to automate white collar work, a lot of which are these administrative positions, and then a lot of which are also a great image from a recent Sequoia Capital piece, which is that this is a list of service positions ranked by, if you multiply employees times wage per employee, you get to see the TAM essentially of the different professions. So this is kind of an intense thing, but it's like nurses, software developers, lawyers, accountants, customer service reps. These are people that do white collar work, that do service work, that are ripe to be automated. And so this is why software developers and lawyers and accountants, why there's so much AI pushing to try to be a co-pilot, but also fully automate some of these positions away. And you'll see things like some of these automated nursing platforms where instead of paying for a normal nurse, because we're already paying $284 billion for normal nurses, maybe instead you can pay for some AI nurse. Which sounds dumb, but also my real doctor did miss a blood test recently, but my AI doctor got it right. So there's that. So in the long run, AI is likely to model the whole biological system from the bottom up, from proteins and atoms all the way up through cells and multicellular systems and organs. And it might take a bit of time to get there. Until we do, we believe in Derek Lowe, who says that it's a data problem and a complexity problem. But in the meantime, we're going to get a lot of productivity benefits from automating and helping us with some of the white collar work in medicine and healthcare. Thank you, as always, for watching. And thank you to Roots donors for supporting this channel. If you want to understand more about the past and how we got here, check out the previous videos here. And if you want to understand the future and where we're going, feel free to subscribe here. Hope to see you then. Bye.